Good afternoon. Can you hear me? चला सगळ्यांना मेसेज द्या. Okay, let us start with interactions bacteria. So we have seen interactions with plants. That was the first point. In that we had covered legume rhizobium symbiosis in root, stem and leaf. So legume rhizobium symbiosis in root nodulation, legume rhizobium symbiosis in stem nodulation, and legume rhizobium symbiosis in leaf nodulations. The second plant interaction was mycorrhiza. So plant root interactions with fungus. That was the part we covered. In that we have seen ectomycorrhiza, endomycorrhiza, orchid mycorrhiza and arbuscular mycorrhiza, 4 types. Then we have seen again one more plant interaction, that is lichen, a mutualistic symbiotic association between algae and fungi. Then the last one we have seen is PGPR, plant growth promoting rhizobacteria. So these are a group of bacteria which enhance the growth of the plants either by providing nutrients or by inhibiting the growth of phytopathogens. So these were the interactions in plants. Then in interactions in animals, we have seen ruminant symbiosis. So how ruminant animals, they show a specific type of digestive system involving 4 parts of the stomach, that is reticulum, rumen, omasum and abomasum. So how the food or cellulose is digested in their intestine with the help of cellulases. The next interaction is interaction of bacteria. That is, in interaction of bacteria, we are going to cover bacterial bioluminescence. So these are the images of 2 types of fishes which show bioluminescence. So this pincone fish, it utilizes luminous bacteria, and those are colonized, or they form colonies in the ventral cavity. So this ventral part of this pincone fish, it shows this type of illumination. So this illumination will help to spread light in the surrounding area because these fish are found in water where it is very dark. Sunlight cannot reach the depth of the sea. So in those regions wherever there is dark, to illuminate that region, to create light in that region, they swallow luminous bacteria. They utilize luminous bacteria that reach their ventral cavity, and in that cavity, these luminous bacteria have the ability to show bioluminescence, and that bioluminescence, that glow, that shine, produces a sort of light, and that helps them to illuminate the surrounding area where these fishes are present. This is also used for intra-species communication, to communicate with the mating partner. This is an angler fish. It carries luminous bacteria in a light emitting rod. So there is a rod like structure. Images this type. This is a rod like structure. Okay. And this is the ventral cavity of this pincone fish. In this ventral cavity, it takes inside luminescent bacteria which show that typical blue type glow, and that helps to create illumination, or that helps to illuminate the surrounding area where pincone fish is present. And this one is the to show you a bigger image. So this is a deep sea angler fish. Okay, can you see it now? Very clear it is deep sea angler fish. Now, what is the characteristic of this fish? It has a typical rod-like structure, and this rod-like structure So, because of this luminescence, the smaller fishes or the prey is attracted towards this fish, and as they are attracted towards this light, this fish can engulf them or eat them. So, this is how the parasite prey, predator prey relationship is found in this fish because of this bioluminescence. Wait a second. Okay. So, anglerfish and pincone fish. So, luminescent bacteria, they are light emitting microorganisms, and they usually are found in the sea water as well as in some of the terrestrial water also. Water also. Many species of luminescent bacteria, they can exist freely, and they are usually associated in the sea, such fishes, squids, crabs, nematodes, etc. In symbiosis, the bacteria, they are nourished with the readily available food from the fish, and at the same time the host utilizes the bioluminescence of this bacteria for communication, for attracting the prey, and to protect itself from the predators. समजा त्या फिश खूप छोट्या आहेत, तर त्या लाईटमुळे मोठ्या फिश घाबरून जातील की ते काहीतरी आहे. सो दॅट इज नोन ॲज मॅस्करडे, टू प्रोटेक्ट देमसेल्स फ्रॉम दी प्रिडेटर. म्हणजे त्यावेळी ह्या फिश प्रे असते. Is that clear? See how interesting this relationship is. So symbiotic bacteria, what they do is they get the readily available food material from the fish, and what the fish get in return, they use the bioluminescence property of this bioluminescent bacteria, and that bioluminescence, they will help in different tasks for the fish. कोणकोणत्या कामासाठी ते युजफुल असू शकतात? टू कम्युनिकेट, ओके, फॉर mating with the partners, for breeding purpose. They communicate with their fellow fishes. So तिथे ते अट्रॅक्ट करू शकतात. सेकंड, आपल्या प्रेला अट्रॅक्ट करू शकतात, की प्रेला वाटेल अरे इथे काहीतरी खायचं आहे, मग गेलं की लगेच त्या फिश त्याला खाऊ शकतील. मोठ्या असतील तर छोट्यांना खातील, आणि छोट्या असतील तर ते त्या गोमुळे मोठ्या फिश घाबरून जातील की अरे इथे काहीतरी असेल तर, म्हणून ते तिथून दूर. मॅस्पर्गेट मीन्स टू हाईड देमसेल्स फ्रॉम दी प्रेडेटर्स, म्हणजे त्या ग्लोच्या मुळे मागे फिश आहे तर ती हाईड होऊ शकते त्याच्या मागे. सो दीज आर दीज uses of this bioluminescence. There are certain species of luminescent bacteria which are obligatory symbionts, आणि कंपल्सरीली ते त्या फिशमध्ये दिसतील म्हणजे दिसतील इन symbiotic association, and they require nutritional their nutritional requirement will be completed by the host. These obligatory symbionts are not separable for the host from the host. They cannot be separated from the host and are unable to be cultured in the laboratory. So if it is this bioluminescent bacteria, if they are obligatory hosts, then they cannot be separated from the fish. And if we try to separate them and isolate them in the fish, आपल्याला वाटेल की किती छान ते ग्लो दाखवतात, तर ते बॅक्टेरिया जर आपण आयसोलेट केले आणि लॅबमध्ये ग्रो केले तर ते काही बायोलुमिनेसेन्स दाखवणार नाही आणि ग्रो पण होणार नाही. So that type of obligatory symbionts are very difficult to be cultured in the laboratory conditions. Now there are three major genera in which these luminescent bacteria are classified. One is Photobacterium, another one is Vibrio, and third one is Photorhabdus. So these three genus include the bioluminescent bacteria. Marine environment usually contains Photobacterium and Vibrio, while terrestrial species are included in Photorhabdus genus. आधी त्याला झिनोरॅप्डस जीनस म्हटलं जात होतं, आता त्याला फोटोरॅप्डस असं नाव दिलेलं आहे. सो हिअर you can see in this diagram, luminescent bacteria, they are present in the special organs which are found in this region, बाहेरच्या कवरिंग रिजनच्या mantle part मध्ये हे luminescent bacteria आहे. त्याच्यामुळे the fish is showing the typical glow in the mantle region. Okay, and this bioluminescence or luminescent activity in the bacteria, it is because of the enzyme known as luciferase. So this is the structure of that luciferase. It has two subunits, alpha subunit and beta subunit. हा त्याचा अल्फा सबयुनिट आहे आणि हा त्याचा बीटा सबयुनिट आहे. हा जर luciferase enzyme नसता, okay, then it was very much impossible for the luminescent bacteria to show the bioluminescence property. Some luminescent bacteria, they are parasitic. For example, Photobacterium and Vibrio, they can infect the crustacea group of animals, and Photorhabdus, they usually infect the terrestrial insects such as caterpillars, nematodes, and they use them as a host. The free living luminescent bacteria can often be found in the gut and on the skin surface of marine animals, and they are not parasites. Actually, those which are present on the surface of the skin or in the intestinal region or in the lateral region or ventral region, they are just in symbiotic association, and they are non specific parasites. Means they are not parasitic on that fish, they are only in symbiotic association. पण काही luminescent bacteria जे आहे ते parasitic असतात आणि ते permanently त्या fish करू शकतात. For example, Photobacterium and Vibrio, as well as Photorhabdus luminescens. So Photorhabdus luminescens, usually it attacks the terrestrial insects such as caterpillars and nematodes. Let us see why this luminescence is observed. What is the biochemical or what is the reaction involved in this bioluminescence? So that is because of, I already told you, it is because of the enzyme luciferase, bacterial luciferase. So bacterial luciferase is the enzyme that catalyzes the light emission and shows the bacterial bioluminescence. It is the enzyme which carries out the typical reaction required to produce that light. The catalytic machinery involved in continuous light production in luminescent bacteria includes not only bacterial luciferase but also enzymes that supply and regenerate the substrate for bacterial luciferase. आता luciferase हा एन्झाईम आहे. तर एन्झाईम कशावर अटॅक होईल तर तुम्हाला लाईट प्रोडक्शन मिळेल तर त्याचा सबस्ट्रेट कुठला तरी असेल, कुठला तरी केमिकल कंपोनंट असेल त्याच्यावर हा लुसिफरेज एन्झाईम अटॅक करत असेल, त्याची कॅटॅलिटिक ऍक्टिव्हिटी दाखवत असेल आणि त्याच्यामुळे फोटॉन्स ऑफ लाईट इमिट होत असतील तर असा सबस्ट्रेट प्रोव्हाइड करायचं काम सुद्धा या बॅक्टेरियाचे असतात. सो दे हॅव टू कलेक्ट दॅट सबस्ट्रेट विच इज रिक्वायर्ड फॉर the bacterial bioluminescence. The DNA sequences coding the proteins in luminescent system are termed as lux genes. So lux genes म्हणजे तो लक्स साबूनशी काही संबंध नाही. जे जीन्स लुसिफरेज एन्झाईम तयार करतात त्यांना तुम्हाला लक्स जीन्स असं नाव दिलेलं आहे. बॅक्टेरियल लुसिफरेज इट इज अ हेट्रोडायमर. इट हॅज 2 युनिट्स. वरती आपण डायग्राम मध्ये इथे पाहिलं 2 युनिट्स आहेत इथे, दिस वन अँड दिस वन. सो इट इज अ heterodimer. Hetero means both the units are different. That is why we call it as hetero, and dimer because it has 2 units. So there are 2 polypeptide chains in bacterial luciferase or this heterodimer. One is alpha chain and another one is beta chain. The alpha chain has a molecular mass of 40 kilodaltons and beta chain has molecular mass of 37 kilodaltons. Both of these alpha and beta chains, they are coded by Lux A and Lux B genes. I already told you, luciferase enzyme तयार करण्याची क्षमता या बॅक्टेरियामध्ये का असते? कारण त्यांच्याकडे तसे जीन्स आहेत. So those genes are known as Lux genes. So Lux A gene synthesizes the alpha subunit of the luciferase, while Lux B gene synthesizes the beta subunit of the luciferase enzyme. The substrates for this bacterial luciferase are flavin mononucleotide FMNH2, molecular oxygen, and long chain fatty aldehydes. So all these are carbohydrate type of components. So it is one is the vitamin flavin mononucleotide, molecular, it's a growth factor, oxygen लागतं आणि long chain fatty aldehydes लागतात. So derivatives of fatty acids. Excess energy which is liberated from flavin mononucleotide and aldehyde concomitant with the reduction of molecular म्हणजे काही एनर्जी रिलीज होते कोणाकडून फ्लॅविन मोनोन्यूक्लियोटाइड कडून आणि लॉंग चेन फॅटी अल्डिहाइड कडून. ती एनर्जी कशासाठी वापरली जाते? टू रिड्यूस द मॉलिक्युलर ऑक्सिजन अँड एमिट द light in the form of blue green light emission having a maximum lambda max or maximum wavelength of 490 nanometers. Is that clear? Once again we'll repeat in short. In bacterial in interactions with bacteria, the first example is bacterial bioluminescence. So this bacterial bioluminescence is a property which is shown by bacteria which contains specific enzymes which have the ability to show the luminescence, that is blue-green type of glow. These 2 images are, one is of pin cone fish. So in this ventral cavity, it utilizes those bioluminescent bacteria from the sea and they reach this ventral cavity and show typical type of glow or blue-green light is produced. So the fish मध्येच ती light सुंदर दिसते एकदम. And this is another type of angular fish, deep sea मध्ये असतं ते. आणि ह्या रॉड लाईक स्ट्रक्चर असतो, ह्याच्यात ते ल्युमिनेसेंट बॅक्टेरिया असतात आणि ते टिपिकल ग्लो दाखवतात. हा ग्लो कुठे कामात येतो? तर एक तर त्यांना त्यांचं प्रेला अट्रॅक्ट करण्यासाठी ते यूज करू शकतात, स्वतःला प्रोटेक्ट करण्यासाठी यूज करू शकतात किंवा मेटिंगसाठी यूज करू शकतात. Luminescent bacteria are found in usually sea water and also some are found in terrestrial or freshwater systems. They may be found as free living in the water or they may be in symbiotic association with fish, crabs, squids, nematodes, etc. So what is the association? They get ready-made food from the fish, and in turn they provide this bioluminescent property to the host, that is the fish. So the host utilizes it for communication, for attracting the prey, and to hide itself from the predators. Some of these are some of these bioluminescent bacteria, they are infectious and they cause infection in these type of marine environment, and those are Photobacterium and Vibrio as well as Photorhabdus. So they when they attack the fishes or insects or caterpillars or nematodes, they cause infection in them. Some of them are freely found in the intestine and on the surface of the skin, but they are not parasites. What is the chemistry behind the bioluminescence? That is bacterial luciferase. So it is the enzyme which uh, carries out this particular reaction and shows the emission of light. So catalytic machinery that is involved in bioluminescence is nothing but bacterial luciferase, and it is nothing but an enzyme with two subunits. One is alpha subunit and another one is beta subunit, having a molecular mass of 40 and 37 kilodaltons respectively. These two subunits are synthesized from two genes, Lux A gene and Lux B genes. So because of the presence of Lux genes, these bacteria can show the properties of luminescence. Now what is the reaction involved? This luciferase, it attacks flavin mononucleotide. So it reacts with, it shows reaction with flavin mononucleotide and long chain fatty acid aldehyde, which are present in this organism. They release energy. This energy is used for reduction of molecular oxygen to produce blue-green light, which has a wavelength of 490 nanometers approximately. The second example of bacterial interaction is microbial kappa particles. So they were first discovered by T.M. Sonneborn, and he found these type of kappa particles in Paramecium aurelia. So it is a sort of cytoplasmic particle, and Sonneborn named it as kappa particles, and normally it is connected with the genetic system. So kappa organism, also known as kappa particle, it is nothing but a, it's earlier it was considered to be particle, so त्याला particle now पडून गेलेला आहे, but actually it is a gram negative symbiotic bacteria found in the cytoplasm of some of the protozoans of, for example, Paramecium aurelia. This bacteria, when released into the surroundings, changes the P particles to secrete the poison. So here, Paramecium ला ते ज्या पॅरामेशियम मध्ये हे बॅक्टेरिया प्रेझेंट असतात त्यांना ते फोर्स करतात टू रिलीज अ पॉयझनस सबस्टन्स दॅट इज पॅरामेसिन that kills the other sensitive strains of Paramecium aurelia. So this is a type of competition between various species of Paramecia. आणि Paramecia aurelia मध्ये हा प्रोटोझोआ आहे, स्लिपर शेप्ड बॉडी असते त्याची, ज्याच्यामध्ये हे कापा पार्टिकल्स प्रेझेंट असतील, म्हणजे हे बॅक्टेरिया प्रेझेंट असतील, ग्रॅम निगेटिव्ह सिम्बायोटिक बॅक्टेरिया प्रेझेंट असतील, त्याच बॅक्टेरियामध्ये ही प्रॉपर्टी असेल टू प्रोड्यूस अ पॉयझनस सबस्टन्स नोन एज पॅरामेसिन. दिस पॅरामेसिन कॅन किल दी strains which do not contain this ability to produce the paramecin or poisonous substance. म्हणजे they are lacking in kappa particles. पॅरामेशियम जर आपण कापा पार्टिकल्स काढून टाकले तर ते काही मरत नाही पॅरामेशियम. The cytochrome pigments utilized by kappa for respiration are different from those of host animals, but they are similar to those present in some bacteria. आता हे कापा पार्टिकल्सची जी ब्रीदिंग मेकॅनिझम आहे किंवा सायटोक्रोम सिस्टीम आहे, तर ती जी सिस्टीम आहे ती बॅक्टेरिया सारखी असल्यामुळे ते बॅक्टेरिया आहे असं आपण म्हणू शकतो. It has been suggested that kappa particles have originated independently from paramecium in the distant past. जसं असेल की अं, एक इंडिव्हिज्युअल ग्रॅम नेगेटिव्ह बॅक्टेरिया म्हणून तो एक्झिस्ट होत असेल, पण पॅरामेशियाच्या बॉडीमध्ये तो चुकून एनगल्फ करताना शिरला असेल अँड इट हॅज बिकम अ परमनंट मेंबर ऑफ पॅरामेशियम ओरेलिया प्रोटोझोआ. म्हणजे इव्होल्युशनच्या वेळी तो पार्टिकल पॅरामेशियममध्ये शिरला असेल अँड इट हॅज एंटर्ड इन अ symbiotic relationship or as an endosymbiont in paramecium aurelia. These kappa particles appear bright or non-bright under light microscope. आपण मायक्रोस्कोप मध्ये बघायचा प्रयत्न केला ते कसे दिसतात, ब्राईट or non-bright. Peer and co-workers in 1971 isolated a virus from the bright kappa particles. बघा आता, म्हणजे पॅरामेशियम हा प्रोटोझो आहे, त्याच्यामध्ये हा ग्रॅम निगेटिव्ह बॅक्टेरिया और कापा पार्टिकल आहे, त्याच्यामध्ये सुद्धा व्हायरस आयसोलेट केला गेलेला आहे. व्हायरस कुठे कुठे प्रेझेंट आहे ते बघून घ्या. व्हेरी डेंजरस. We very well know today how these viruses are dangerous. So Preer and co-workers in 1971, they have isolated a virus from these kappa particles. These kappa particles contain proteinaceous R bodies and they are responsible for the toxic effect or paramecin effect of these kappa particles. So paramecin release करण्यासाठी हे R bodies essential आहे. We can see these kappa particles in paramecium. This is the paramecium and this orange कलरचे जे छोटे छोटे दिसत आहेत ते आहेत कापा पार्टिकल्स. पर्पल कलरचे राइबोसोम्स आहे. दिस इज बिग वन. Big orange yellow part is mitochondria. This is micronucleus, and this is used for engulfment. ओके, इथून एनगल्फ करतात आणि हे जे छोटे छोटे ऑरेंज दिसत आहेत, ओके, थोडीशी करते, थांबा, enlarge this diagram. Can you see now? Okay, these small orange, yellowish orange particles, these are kappa particles. These purple ones are ribosomes. This is mitochondria, micro and macronucleus, and this is the mouth which is used for engulfment. So this is all about the kappa particles. Next one, lichens. आपण ते मिस झालेलं होतं वरती, ऍक्च्युअली टाइप्स ऑफ लायकेन्स बघितलेले होते. सो देर आर 3 टाइप्स ऑफ लायकेन्स. वन इज क्रस्टोस लायकेन्स, फोलियोज लायकेन्स अँड फ्रुक्टिकोज लायकेन्स. सो लायकेन्स आर ऑफ 3 टाइप्स. अल्गल अँड फंगल असोसिएशन आपण बघितली, तिथे हा पॉइंट मिस झालेला आहे. So crustose lichens, these are usually found on the crusts of the rocks, soil crust, barks of the tree and the roofs of the houses. Mostly they are found in grayish green color or they may be red or yellow in color also. So rough patchy surface तयार करतात ते, जसे ते झाडाचं थडचं बार्क असतं, म्हणजे वरचं लाकडाचा पार्ट कसा खरबड असतं तसंच ते क्रस्टोस लायकेन्स आपल्याला दिसतात. फोलियोस लायकेन्स आर फ्लॅट कन्व्हर्टेड इन अ लीफी फॉर्म, म्हणजे पानासारखे दिसतात म्हणून फोलियोज lichens. Most of them they grow on the upper and the lower surface of the leaves. Fructicose are hair-like structures, दोऱ्यासारखे दिसतात. Mostly found hanging from the trees, असे झाडाच्या वरून ते हँग करतात. They may also be present on the upper or lower surface of the leaf. Ecological and economic importance of lichens. So lichens, तुम्हाला माहिती आहे, it's a compulsory type of association. समजा हे चुकून लायकेन्स मरून गेले तर तो जो डेड पार्ट असतो लायकेन्स, इट इज व्हेरी रिच इन मिनरल्स अँड दॅट कॅन बी युज्ड फर्दर for various purposes. In tundra regions, lichens are found in large quantities. They are used to enrich the soils and to entrap the dust, water, and pollutants. When lichens die, they contribute to the organic matter of the soil. जर lichens मरून गेले तर जिथे ते केले तिथली सॉइल रिच इन ऑर्गॅनिक मटेरियल होऊन जाते. दिस इम्प्रूव्स दी फर्टिलिटी ऑफ द सॉइल. They are also important in nitrogen fixation because algae is one part and algae is involved in uh, this type of association. So they can convert nitrogen from the air into nitrates, and this is very important in the increasing the nitrate contents of the plants. Lichen needs clean air to grow. They are not able to tolerate pollution. So they can absorb everything. If to grow, they absorb everything from the air, including carbon dioxide and heavy metals. So scientists have determined the level of air pollution in an area with the help of lichens. If lichens die, if you take some lichen to a city area and if it dies, means it is very polluted area, and so the lichen could not survive there. So this is a signal or a warning that the pollution of that particular region is increasing drastically and it is very harmful for human beings. हे आपण बघितलं होतं. Perfumes can be made from lichens. So specifically त्याची example आहे Ramalina lichen. Lichens like stone mushrooms are used as food. They are also chemical substances. They are used in medicines. They contain some chemical property. So microbial dosage of sulfur dioxide pollutionचा प्रॉब्लेम सॉल्व करू शकतात हे lichens. This is all. We have finished unit 2 today. That clear? सेकंड युनिट झालेला आहे आपण. नेक्स्ट लेक्चर पासून we will be starting the last unit, third unit. Very complicated one. पहिले 2 युनिट सोपे सोपे होते. तिसरा युनिट थोडासा कन्फ्युजिंग आहे. So let us stop here. तुम्हाला काही विचारायचं असेल तर सांग. Want to ask anything? Ask. Want to ask anything? कोणाला काही डिफिकल्टी आहे? अदनान, प्रीति, निकिता, विक्की, any difficulty? काही विचारायचं असेल तर विचारा, मी थांबवते. ओके. नेक्स्ट युनिट सुरू करू आपण मंडेला, लास्ट युनिट. ओके. याचे नोट्स मी दिलेले आहे, कंप्लीट करून ठेवायचं आणि रीडिंग करून ठेवायचं. क्वेश्चन्स देईल मी तुम्हाला.